The overall goal of this procedure is to analyze the cytokine secretion, transcription, and morphological impact of stimulated T cells. First, use a Phi-Col gradient to isolate the peripheral CD8-depleted blood mononuclear cells from the HIV-infected individuals. Incubate the cells with either the isotype control or the blocking antibody of interest. Then stimulate the cells with the antigen of interest. Next, centrifuge the cell cultures in order to collect supernatants for cytokine measurements and cell pellets for phenotypic analysis or transcriptional analysis. If desired, adapt this protocol to include sorting the individual cell types by flow cytometry. Results on cytokine secretion, phenotypic, or transcriptional analyses can show the impact of antibody blockade of inhibitory pathways on restoring the effector function of HIV-specific CD4 T cells.
This in vitro assay can be effectively used to quantify cytokine levels restored by the blockade of inhibitory pathways, to investigate a broad panel of effector molecules, and also to evaluate the impact of cytokines produced by HIV-specific CD4 T cells on other cell subsets. Meghan Hart, a research technician from our laboratory, will now demonstrate this procedure.
Deplete the CD8-positive T cells by adding the human CD8-positive depletion cocktail at 50 microliters per milliliter of whole blood. Mix well and incubate for 20 minutes at room temperature. Mix the whole blood with Hanks buffer. Now carefully layer the blood over histopac. Then centrifuge the gradient at 540 RCF for 30 minutes. Collect the layer of peripheral blood mononuclear cells. Transfer the cells to a new 50 milliliter